Now our last topic that we're going to cover for exam 1 is going to be bacterial nutrition and growth. So all living things, so I'm talking about bacteria, I'm talking about humans, need to take in nutrients and other chemicals that act as building blocks to make molecules. So some of the most common nutrients that we need and that bacteria need to take in are necessary elements or building blocks to make other molecules. So the most common ones are going to be carbon, hydrogen, nitrogen, and oxygen. We'll start off talking about oxygen as a chemical requirement for growth. And oxygen is an essential for growth. Or is it? Remember in the last lecture when we were talking about metabolism, all organisms actually don't need oxygen in order to get energy. So humans need oxygen. We're obligate aerobes, but all types of bacteria do not need oxygen. So bacteria can be obligate aerobes and require oxygen. They can also be obligate anaerobes and actually die in the presence of oxygen. I showed you a similar image before with these thioglycolate tubes and where bacteria would grow in a tube. I'm just pointing out again the difference between oxygen requirements of organisms. All of the oxygen is at the top of the tube. This is where all obligate aerobes would grow. And then there's no oxygen at the bottom of the tube. This is where obligate anaerobes would grow. So is oxygen absolutely necessary for growth? No, it depends on the organism. But the oxygen requirements must be met for that specific type of organism to grow. So if it needs oxygen, oxygen needs to be present. If it dies in the presence of oxygen, then it needs to be in a very low concentration or no oxygen environment. Our next chemical requirement for growth after oxygen, now we have nitrogen. And nitrogen is a very important building block because nitrogen is in a lot of organic compounds. So we'll see a couple examples on the next couple slides. Example 1, does anybody know what these are? And I'll save you from counting them. There are 20 of them. And did you guess correctly? So these are the 20 amino acids. So there is an amino group in all amino acids. And look, it's circled in each one. There's a nitrogen group in every single amino acid. So nitrogen is really important for amino acids, and amino acids build proteins. So if we don't have nitrogen, we can't build proteins. Next up, we have nucleotide bases. So these are the building blocks for things like DNA and RNA. And look here, we have nitrogen and all of these different types of bases here. So nitrogen is really important for nucleotide bases. Without nitrogen, we would not be able to have DNA and RNA. So nitrogen is a really important chemical requirement building block for growth. Without nitrogen, we wouldn't be able to make proteins, we wouldn't be able to make DNA and RNA, so we wouldn't have anabolism, so we wouldn't be able to build these molecules. We wouldn't be able to build molecules of protein, we wouldn't be able to build molecules of DNA or RNA. And nitrogen makes up a lot of our cells because, like I said, it's really important for we have so many proteins in our cells that do so many jobs. Same with bacteria, they have so many proteins that do so many jobs. And there is nitrogen in every amino acid that builds a protein. And then also DNA and RNA, which is in all living cells as well. And nitrogen makes up 14% of the dry weight of a cell. That is a lot. And so nitrogen is absolutely essential for the growth of living things. All right. I know what you're probably thinking though. Where is all of this nitrogen coming from? So most organisms are going to get their nitrogen from organic compounds. So from consuming an organic compound, like different types of foods. So fish and eggs and nuts and greens all have nitrogen in them that we can consume and break down and then take that nitrogen to use as building blocks. You can also recycle nitrogen from amino acids or nucleotides that are pre-existing. So most organisms get nitrogen by consuming it or recycling it. But what about all the nitrogen in the air? There is so much atmospheric nitrogen. About 78% of Earth's atmosphere is nitrogen, and about 21% is oxygen. So there's so much nitrogen just hanging out in the air. Why are we not able to use it? So all of that nitrogen in the air, the abundance of nitrogen in the atmosphere, is actually in an unusable form for animals, for people, for plants. First, it has to be helped by nitrogen-fixing bacteria to be changed into a usable form. So the nitrogen cycle is this repeating cycle where nitrogen moves through living and non-living things. You have nitrogen in the atmosphere, it goes down to the soil, interacts with nitrogen-fixing bacteria, interacts with water and plants, and then ultimately animals like humans. So in order to move through this cycle, nitrogen has to change forms. So you have atmospheric nitrogen that is then fixed by prokaryotes, or this bacteria in the soil that converts it to ammonia. Ammonia can be taken up by plants, or it can be converted into nitrites and nitrates. And nitrites can be picked up by plants. And then it's this form of nitrogen that humans are going to eat, that animals are going to eat. They're going to eat this form that was taken up by the plant, and then maybe an animal eats it, and then we eat that animal. That's how this altered form of nitrogen is moving through the cycle, and we're able to receive this altered form. So some bacteria, not all bacteria, are nitrogen-fixing bacteria, and they can transform nitrogen gas in the atmosphere into a usable form for other organisms. And this is absolutely essential. If we did not have bacteria fixing the nitrogen in the atmosphere, we would not be able to be living organisms because we would not have the nitrogen to build proteins and make DNA and RNA. So it's an absolutely essential cycle for living things. And now here's some other important chemicals. We have phosphorus, sulfur, calcium. Phosphorus is essential for the phospholipid membranes, like the phospholipid bilayer. It's also an important part of DNA, RNA, adenosine triphosphate, ATP, other types of proteins. Sulfur is important for certain types of amino acids, for certain types of vitamins. But you should be wondering, why are these compounds so essential? And that's because bacteria can't make them on their own. So they have to get these chemicals from their environment. They aren't able to make them up themselves if they don't have access to them. They have to get this stuff from the environment. So because bacteria aren't just able to make it when they need it, these are called growth factors. So these are kind of limiting factors for growth. If they're not available to the organism can't live and grow. They have to be available to the organism in their environment. So I changed this right here to growth factors. They're not just some extra important chemicals. They are called growth factors, and they will limit the growth of an organism if they're not available. And now we're going to talk about some physical requirements for growth. So for chemical, we really focused on oxygen and nitrogen. But remember, the other two that I said were essential were carbon and hydrogen. We really already talked about those when we talked about metabolism. How carbon is an essential building block for complex and more simple molecules, and it holds a lot of energy between those bonds. And then hydrogen is important for transferring electrons from place to place, and hydrogen bonding for holding things together like proteins. So now we're into physical requirements for growth though, and the first one we'll talk about is temperature. And when discussing what temperature does to cells, we're really thinking about proteins and lipids. So this should look familiar. In terms of temperature for proteins, temperature that's too high will denature proteins. So it will change this nice, perfect 3D shape that the protein had. It will break apart the hydrogen bonds that are holding it together, denature it, and make that protein non-functional. So temperature requirements for growth. Temperature needs to be within a certain range for organisms to be able to grow effectively. Otherwise, all their proteins will just fall apart and be ineffective. For lipids, think the phospholipid bilayer of the membrane, or think of fats. When fats become really cold, they get very rigid. So the same concept happens to a membrane of a cell. Whenever a cell becomes too cold, that membrane becomes very rigid, and then it can easily break and then just release the contents of the cytoplasm and die. It can also become very fluid if the temperature gets too high, so the membrane can kind of start to fall apart and disassociate. But most often, high temperatures are going to denature proteins before it makes the membrane too flexible. So hot and cold temperatures influence lipids, but mostly we're thinking cold temperatures for the rigidity of the plasma membrane. So temperatures that are too high will denature proteins and destroy that three-dimensional shape. Temperatures that are too low will make lipids in the membrane too rigid, causing that membrane to break. So knowing those things now, you understand that organisms have a certain range of temperature where they can survive. Otherwise, things just start falling apart and not working. So the minimal growth temperature is the lowest temperature that an organism can still go through metabolism. And lots of organisms and microbes can actually survive below their minimal growth temperature. It's just they're not going to be thriving. Their metabolic activity is going to be way too slow for them to actually grow and replicate. That's what's happening whenever you put stuff in the fridge to keep it fresher, to prevent microbial growth. You're putting it in the fridge so those microbes slow down and stop replicating. They're not necessarily going to die in the fridge, but they're going to stop replicating. On the other hand, maximum growth temperature is the highest temperature where an organism can still continue metabolism. And exceeding this temperature leads to cell death, because at this point, high temperatures are going to damage and denature those proteins. That means the organism can no longer go through metabolism, and ultimately that leads to cell death. And then right in between those two, we have the sweet spot of the optimum growth temperature. So this is the temperature range where the organism's metabolic activity is at the highest rate. So it's making the most energy, it's growing most effectively. And we can categorize microbes based on their optimum temperature ranges, and that's what we see on the next few slides. So this chart is showing different optimal growth temperatures for different types of bacteria. So we have psychrophiles and psychrotrophs, mesophiles, thermophiles, and hyperthermophiles. And you can see on the y-axis we have the rate of growth, so how effectively are these organisms growing, how effectively are they going through metabolism. Then on the x-axis we have temperature in degrees Celsius. And right here for mesophiles, I want to point out that 30 to 40 degrees Celsius is about 95 degrees Fahrenheit. So we'll see what that means. So first off, we're going to kind of lump these together and talk about psychrophiles and kind of psychrotrophs in one. But these organisms thrive in these lower temperatures. So right here around, 10 degrees Celsius, that's about 50 degrees Fahrenheit. And that's pretty chilly. So these organisms grow best at lower temperatures. They can even grow below freezing and still thrive. And a lot of these way down here are going to be more like archaea, so they're not going to cause disease in humans or anything like that. But they love to live in cold environments, so you can think of snow and ice. Most of these organisms aren't going to cause disease in humans because this is such a low temperature. They're not going to be able to thrive inside of a human body, but they can still cause food spoilage in the refrigerator because your fridge is cold, your freezer is cold, and these organisms can still thrive in those low temperatures and continue replicating even in the fridge. An example of a very relevant one is this Listeria monocytogenes. And this is relevant to humans. Even though it thrives in low temperatures, it can still cause food poisoning in humans as well. On to mesophiles. And mesophiles grow best right in this temperature range between 30 and 40 degrees Celsius. So this would be about 95-ish degrees Fahrenheit. And that is just about body temperature. So these types of organisms grow best at human body temperature. And that should tell you that these organisms are the ones that are most likely to cause human diseases. So most organisms that cause diseases in humans belong to this optimal growth temperature group of mesophiles. And then finally we have thermophiles and hypothermophiles. And these organisms grow best in high temperatures. They really start popping off here at about over 120 degrees Fahrenheit, so that is very hot. And then when we get into hypothermophiles, they start really thriving around 200 degrees Fahrenheit. So they thrive in really hot conditions like hot springs. And these, of course, are going to be mostly archaea. So they're going to be extremophiles. They're going to live in extreme high heat conditions. And these are really not going to cause diseases in humans because they would not be able to thrive and survive in the human body temperature that's way down here. So our next physical requirement for growth that we'll talk about is pH. And this stands for potential hydrogen, and it tells us how much hydrogen is essentially in a liquid. And you've probably seen this scale before. pH is really measuring how acidic or basic a substance is. So lower numbers on the pH scale are more acidic, and then higher numbers on the pH scale are more basic or alkaline. And like I said before, this really has to do with hydrogen. So the concentration of hydrogen ions, a positive charge, or hydroxide ions, a negative charge. Organisms are really sensitive to changes in acidity and pH because their proteins that are keeping that three-dimensional shape that's so important for their function are held together by hydrogen bonding. So all of these charged hydrogens can kind of run around and rip off hydrogens and change the shape of those proteins. So generally speaking, most types of bacteria and protozoa are going to thrive best in this neutral range on the pH scale. Now, of course, there are plenty of archaea and types of extremophiles that can exist in these more extreme ranges on the pH scale, but generally, they like it to be neutral. And surprise, surprise, most of the organs of the human body, the tissues and organs, are all within this neutral range as well. So this is also the preferred range for pathogens. Those are organisms that cause disease in humans. So pathogens that can get inside us and start replicating and infecting us and causing disease thrive in this neutral range, which makes sense because the human body is also mostly neutral. So environments that are too acidic or too basic will inhibit microbial growth. And there are organisms that produce acidic waste byproducts. Those acidic waste byproducts accumulate in the environment and inhibit growth of bacteria that cannot survive in an acidic environment. So an example would be fermenting bacteria and fungi that are used to make cheeses. So these types of bacteria produce lactic acid, which is acidic. The lactic acid builds up in the environment, lowers the pH of the environment. The lactic acid bacteria doesn't mind, though, because it thrives in that environment. But other types of pathogens that would normally make a home on this cheese now cannot make a home on that cheese because it's too acidic. So it's a way to preserve cheese from pathogenic bacterial growth. Very, very, very interesting. Although most of the human body, tissues, and organs are neutral, there are some areas of the body that are acidic. So we all know the stomach is acidic. We've heard of stomach acid. And in most cases, the stomach acid does limit the amount of microbial intake. So you'll eat something, it may have pathogens or just a lot of microbes. Stomach acid will break down and get rid of those microbes. But there are certain types of acid-tolerant bacteria, like Helicobacter pylori, and we'll talk about this more later on in the semester. But this type of bacteria causes stomach ulcers, and it does that by actually neutralizing the stomach acid and wiggling its way into the epithelial cells of the stomach. Another example of an acidic location of the body is the vagina. So the vagina actually has this resident lactobacillus bacteria. It's this lactic acid producing bacteria. And just like we saw with the cheese, It produces lactic acid and makes a more acidic environment where bad bacteria can't grow. And that's fantastic because it really helps the microbial community stay in check in the vagina. Now, whenever that microbial community gets thrown off, maybe by taking some type of antibiotic, it kills off a lot of the bacteria that is creating lactic acid and making a more acidic environment. Now you have an opportunity for pathogens to come thrive because it's a more neutral pH. And this is called dysbiosis, whenever a microbial community is thrown off and it's outside of the realm of being healthy for that certain tissue or organ. As we saw with temperature, also with pH then, organisms are sensitive to changes in acidity. Environments that are too acidic or too alkaline are going to kill most bacterial cells or microbes. And for the most part, the optimum pH range for organisms and microorganisms is near neutral. We did talk about some examples of organisms that exist out of that range, but for the most part, it's going to be neutral. And that's including pathogens as well, because the human body tissues are mostly neutral. Pathogens that get into us and cause disease are going to thrive in a neutral pH as well. On to biofilms. And I love biofilms. These aren't movies about biology, even though I'm sure those would be interesting too. Biofilms are actually complex microbial communities. So microbes have come together and they work together to create a really resilient environment where they can thrive more effectively, which is good news for them, not so much good news for us, as we'll see. Biofilms are everywhere. So biofilms are extremely common in nature. So if you've ever stepped into a little stream barefoot and the rocks are all slimy, those are biofilms. If you've ever noticed that your shower curtain is looking a little dank, that's covered in biofilm. The plaque that you have on your teeth and you go to the dentist and they scrape it all off, that's biofilm. And then biofilms can also form inside of our body, like inside of our gut. It can also form on lots of different types of medical equipment, like contact lenses, urinary catheters, prosthetic joints, IUDs. And this is a really big problem because biofilms, like I said, are really resilient. They're really hard to get rid of. So biofilms forming on these devices that are going to be inserted into your body or have access to a sensitive part of your body can easily lead to an infection. In the lab, you're going to grow a lot of types of bacteria, but you're going to know exactly what type of bacteria you're growing, and only one type of bacteria is going to grow on a plate. So maybe it'll be Klebsiella, it'll be E. coli, it'll be Staphylococcus, and your plate will be beautifully labeled. But that is not how things are happening in nature. In nature, there's so much going on, there's so many organisms, and so they're living in association with all of those other organisms. So microorganisms in nature primarily reside in biofilms, or in these communities with other microorganisms. One study even showed that more than 10 billion bacteria per square centimeter formed on those slippery stream bed rocks I mentioned on the previous slide. And then the CDC estimates that the biofilm causes up to 70% of bacterial diseases in industrialized countries. The one I want to bring your attention to right here are nosocomial infections. So these are infections that you would get from the hospital during a hospital stay. And these are associated with implantation medical devices. Most often this is going to be from urinary catheters causing urinary tract infections. And that's because these biofilms are really difficult to get rid of. So when a biofilm forms, it's really difficult to just wash off and get rid of. We'll talk about how there are certain steps involved with actually preventing biofilm formation and getting rid of them, but they are extremely resilient. How are biofilms forming? So we start with a free-living cell or a cell in a planktonic state. That cell is then going to adhere or attach to a surface. Maybe it's using its fimbriae or glycocalyx, if you remember that from a previous lecture. At this point, it's going to start developing this extracellular matrix, and it has a bunch of other stuff in it, like proteins and DNA. It's just a tangle of things. And then it's going to start maturing. And this sticky matrix kind of forms all over this microbial community and sequesters certain things in terms of nutrients and certain types of bacteria. And it really organizes itself into a very effective community. At that point, this mature biofilm can disperse some of the bacteria. Some bacteria can just kind of break off and disperse and become free living again, and they can go form a biofilm somewhere else. And this biofilm can just keep thriving here as well. Bacteria are working together to form this biofilm by quorum sensing. So this is essentially bacteria communicating with each other, sending messages to each other to say, hey, you want to form a biofilm? Sure, I'm in, I'll join. Quorum sensing plays a really important role for starting the formation of a biofilm and then continuing the development into a mature biofilm. When bacteria wants to form a biofilm, it will turn on those genes in its genome to start forming that biofilm and secreting quorum-sensing molecules. So these are the molecules that microorganisms have to secrete in order to respond and communicate with other microbes that are around. So quorum-sensing molecules in the environment tell microbes about the density, so how many other microbial cells are around, and the diversity, what other types of microbial cells are around. And this lets the bacteria know, hey, do I want to make a biofilm with you guys or not? As the concentration of the quorum-sensing molecules increases, then that tells the microbes around, yes, everyone's in, we're all making a biofilm, and that's when they will start attaching to the surface and expressing those genes to actually secrete the matrix and start developing into that more mature biofilm. As I mentioned, biofilms are pretty hard to get rid of, which makes them pretty cool because it's bacteria communicating with each other to make this really complex environment that's really resilient. But it's not great news for us, especially in the term of like healthcare and human disease, because they are really hard to get rid of. So a big question is, how do we get rid of them, or how do we stop them from forming in the first place? There are drugs available that can actually block the receptors on the surface of bacterial cells so they can't see other quorum-sensing molecules. So although bacteria are saying, hey, I'm ready to form a biofilm, they aren't picking up those molecules and sensing it, so they're never forming a biofilm in the first place. Another way that you can help your body get rid of bacteria before it starts forming a biofilm, this might sound strange, but you can actually amplify the quorum sensing. So you can increase the number of quorum sensing molecules. And why would you want to do that? Because that alerts your immune system to, hey, there is a pathogen present, and now your body is aware. It can clear the invaders before they even start forming a biofilm. So the increased number of quorum sensing molecules can alert your immune system to an infection before it gets worse. Now, bacteriophages is a way that you can get rid of biofilms. Bacteriophages are viruses that specifically infect bacteria. And this is an image of a bacteriophage over here. And you can engineer biofilms, so you can give it instructions in a lab to go find certain types of bacteria and then inject those instructions into the bacteria that are maybe self-destruct instructions, so it will break down the biofilm. And this is a really useful tool because the sticky matrix on the outside of biofilm is really difficult to penetrate, but bacteriophages actually function like a syringe or a needle, and they'll inject this information into the biofilm. Pretty cool. And then finally, physical disruption as well. So things like sonication or ultrasonication are one way to break apart that sticky matrix on the outside. Because once you've broken apart the biofilm and released those cells, it's easier for your body to get rid of, or for other types of drugs or antimicrobials to get rid of. This is also what's happening whenever you go to the dentist. That's why they have to scrape your teeth. That's physical disruption of the biofilm that is formed on your teeth. The last topic for this lecture is microbial growth. And microbes do not grow like we do. So we started out small, we started out as babies, and then we grew into adults. So our size literally got much bigger, right? That's not how microbes grow. Microbial growth is talking about cell division. So it's talking about one cell turning into 2, turning into 4, turning into 8, and so on. A cell is not getting bigger and bigger and bigger. It's just dividing to make more cells. Microbial growth is referring to an increase in the number of cells, 1 cell to 2 cell to 4 cells, and so on, instead of an increase in size of that cell. So when there's enough microbial growth, you actually see that growth, and we'll see this in the lab. So each of these little white dots on this plate is microbial growth, and it's a visible colony at this point. We can actually see bacteria. Each of these colonies came from one bacterial cell, and we call this a CFU, or a colony forming unit, because this one bacterial cell formed a visible colony. Because there was one cell here, there was one cell here, there was one cell here whenever you put this bacteria on the plate, and then that one cell multiplied, 2, 4, 8, 16 cells, to the point where we could actually see it. And then we have something called generation time. This is also called doubling time. This is the amount of time it takes a population of bacteria to double in number. So the amount of time it takes one cell to become 2 cells, or the amount of time it takes 2 cells to become 4 cells. And this differs in terms of the nutrients available and the environment that bacteria are in. So E. coli can double every 15 minutes in a lab setting. That's so fast. It's more like 15 hours in the wild because it doesn't have access to all of the nutrients right there and the optimal temperatures all the time. But 15 hours is still very fast. What is the generation time or doubling time of humans? If you want to pause here and think about it for a second, you can. But the generation time or doubling time for humans is usually somewhere like 20 to 30 years. So that's the amount of time it takes us to make a new generation, to have a son or a daughter, and then they'll grow up, and in like 20 to 30 years, they'll have children. So the generation time or doubling time of humans is much longer than that of bacteria. So you can see how bacteria is able to evolve and respond to the environment a lot more quickly than humans can. And generation time or doubling time is different for different organisms. So 15 minutes was pretty fast for E. coli in a lab, right? But different types of organisms have it takes them a different amount of time to replicate and make another cell. In some cases, it can be over a day. And for bacteria, that's pretty slow, which is crazy when you're thinking about it in terms of humans taking 20 to 30 years. As bacteria are going through generation time and doubling time, and they're making more of themselves, how are they making more of themselves? They're going through a process called binary fission. Binary fission starts with a parent cell, and then ultimately that parent cell divides into 2 identical daughter cells. And this should look pretty familiar. This should look like a process called mitosis. Mitosis is what multicellular eukaryotes do. Binary fission is what unicellular prokaryotes do. But they are very similar processes. Both start with a parent cell that ultimately divides into 2 identical daughter cells. In terms of binary fission, though, this is a much simpler process than mitosis. Mitosis involves a lot of checkpoints: prophase, anaphase, metaphase, telophase. During binary fission, the circular chromosome, because we're talking about prokaryotes, replicates at the same time that the cell is preparing to segregate and divide. So it's kind of all happening at once very quickly, and then you end up with 2 identical cells. What does doubling time or generation time look like? We start with 1 cell, and the generation time or doubling time of this organism would be 30 minutes. We have increments of 30 minutes. We go 30 to 60 to 90 to 120. So that is the doubling time or generation time of this organism. We start out with 1 cell. It goes through the process of binary fission. We end up with 2 cells. 2 cells divide and then we end up with 4 cells. These 4 cells divide, we end up with 8 cells. These 8 cells divide, and we end up with 16 cells. So this is exponential growth. This is a population growth curve for bacteria, and it's showing the relationship between the number of bacteria and growth over time. On this y-axis, we have the number of bacteria, or really the number of living bacteria, and on the x-axis, we have time. And so during this lag phase, bacteria is just adjusting to the new environment. Maybe you just plated it and you just put it in the incubator. So it hasn't really started growing yet. It's just getting nutrients and preparing to grow. And then you enter the log phase or exponential phase. And this area is where we calculate doubling time or generation time. So this log or exponential phase is when bacteria are exponentially growing. 1 cell became 2, became 4, became 8, became 16, became 32, so on and so on and so on. Eventually, though, we get to the stationary phase, where the number of living cells or new cells equals the number of cells that are dying. So that means there's some type of limiting factor in the environment. Maybe they ran out of nutrients, maybe they ran out of space, maybe too many waste products built up, and they're just not able to create new cells. And then after it has plateaued and you get past the stationary phase, then you have this logarithmic decline or death phase. And this is when all growth has stopped and dead cells continue to outnumber the number of viable cells in the sample. These next several slides just go through the stages of the bacterial growth curve again. They kind of go through what I went over on the last slide. I'm not going to go through it all again, but definitely kind of flip through and look at these in more detail. Make sure you're able to describe what is happening in each of these different phases of the bacterial growth curve.